Thank you. So it's the last talk. I think it's on purpose because I think the organizer wanted everybody to stay till I will give my presentation. No, I'm kidding. So it's work that is financed by the RSLR, and the idea for us is to maybe find new biomarkers or additional biomarkers to monitor ALS. So we use a technology called digital ELISA, which is an ultrasensitive ELISA based on nanobeads. And basically, it's a very classical immunodetection assay with sandwich antibodies. But the idea is really to look individually at nanobeads and to count the nanobeads that are positive or negative. So that's why it's digital. And that's a way like digital PCR to increase the ratio between signal and noise. And therefore, we are 50 to 100 or maybe 500 times more sensitive than regular immunoassays. So we use this technology for NFL initially, and it can be used for many biomarkers, but sometimes when you want to have new biomarkers, you don't have commercial kits, so you have to design the assay yourself, and that's what we do during homebrew assay. It's like for the beer, but here it's for the immunoassay. And basically to do that, we need to first attach capture antibodies on the beads, and then we also need a secondary antibody, a detection antibody that is biotinylated. And when you have that, then you can test different conditions and try to detect your target. And quite often we start with recombinant protein, and you will see what we will do with two of these possible targets. The first one belongs to the neurofilament family, and you heard about neurofilaments, and you have to know that there are many different isoforms in this family, and one of them that is not really well known or used at least is the peripherin. And we had the idea that maybe it's interesting to detect more than just the NFL, because we know that the distribution of these neurofilaments family proteins differs between different types of axons. And it may be interesting to characterize subpopulation or different type of disease to be able to detect a range of these proteins. Moreover, as you know, when you have an axonal damage, you have a release of fragments of these proteins that we can detect both in the CSF and in the blood. So basically, the idea was to look at peripherin as surrogate markers of maybe axonal damage and possibly interesting in different diseases, including ALS. So to do that, first we had to select a range of antibodies, commercial antibodies, that we use for setting up this assay. And in this type of assay, basically, we couple, as I said, a capture antibody on the beads, a biotinylated antibody, and then the idea in this type of experiment, that's just an example, a generic example, we expect when we use a recombinant protein in increasing concentration that we see a signal that is going up with the concentration of the recombinant protein, and obviously we use also different type of controls, either with different antibodies, non-specific antibodies, for example. And that's what we expect when something is working. So I will just summarize what we have done. We have done different type of combination of antibodies, and that's an example with different combinations. Some of them you see they are really flat and they don't work. Some of them they work a little bit, and some of them they seems to be better than the orange here in comparison with the yellow and the green. That's just here. So in this region, you can see clearly that this combination of these two antibodies, 3B3 and 8G2 here, seems to be better. So we developed that on the recombinant protein, but obviously what we need to do next is to do that on biological fluid like blood and CSF from a human being and from patients. And well, it's going to be short here because it doesn't work. We are not sensitive enough to see anything in this fluid with the assay that we developed initially. And just an example here, that's a standard curve. So in the standard curve, you see here that the level is going up. That's what you expect. But when we look at CSF or when we look at blood, basically what we see is just the background. So that was So it did not work yet with these antibodies, and we were disappointed because previously there was a publication showing that with an ELISA that we tried to order, by the way, they saw differences between different diseases. Well, just with a single, with an ELISA like that, to see in the blood such level is a little bit not suspicious, but we need to reproduce that. So we'll see if we can reproduce that. But just very recently, last month, there was this Brain article showing that they could detect peripherin using exactly the same technology as what we did. And they did that in different diseases, including Guillain-Barré syndrome here, but there is no ALS here. And what is interesting is that they use the same antibody as us, but another one. And in this type of assay, the type of antibody that you use is very important. So therefore, maybe it's because they use, it's not because they are better than us, it's just because they use the good antibody. I don't know. But definitely we need to move on with this project. I will talk about that at the end of my presentation. So the second part is related to the detection of the misfolded form of SOD. I mean, you know about SOD is mutated form that is aggregating and that is really involved in the disease, in familial cases especially. And basically, the model is that SOD, the wild-type SOD or the mutated SOD, but especially the mutated SOD, is a change in the conformation and aggregates and is responsible for pathogenic features. And one of the questions that we can also raise is it possible that the wild-type non-mutated is also able to misfold and is present in sporadic cases? And well, obviously this question is important especially because we have therapeutics now targeting SOD. So far it's restricted to the mutated form, but let's say that we can identify sporadic patients with aggregate of SOD that is really confirmed. That is quite important for possible therapeutic treatment. So here we did the same. Basically, we developed an assay for the wild-type non-misfolded SOD, and the idea was to detect the misfolded SOD, and for that we relied on specific antibodies that we obtained in collaboration with Jean-Pierre Julien in Canada, and this antibody is specific for misfolded SOD. And by the way, the results I'm presenting have been just published, most of them in very recently, so you can look at the paper for more details. So then it's always the same idea. We use this folded, normally folded SOD and misfolded SOD, and with this pan-SOD assay, we can really detect folded SOD, and with antibody specific for the misfolded form, we can also detect. So the idea is to see what's going on now if we try detecting the folded SOD with the two different assays. You can see that here the green is the assay for the total SOD, and the signal is much higher than one with the misfolded SOD. You can see that the misfolded assay is detecting a little bit also of the misfolded SOD. That's like this. It's not 100% specific. And when we want to detect the misfolded SOD, it's the opposite way around, meaning that the assay that is designed for the misfolded one is working much better than the other one. So that's already something, and you can imagine that you do the ratio. So we are quite happy to do that, and then we decided to try also samples, human samples, and that's just a pilot study. And in this pilot study, we look at both blood and CSF and blood. And for the CSF, basically the only thing that really we could demonstrate is that, and it has been shown before, is that in sporadic ALS, you have more total SOD, it seems to me, at least in the CSF, than in control. But we could not really see huge differences between misfolded and non-misfolded SOD in the blood of patients, knowing that here it's just like a few patients, and we have not yet analyzed samples from patients with mutated SOD1, and that will be interesting. And we also need to run large assay to really confirm first that we can detect the misfolded SOD1 in the patients with the mutation, and maybe that a few patients in sporadic cases will have some detectable aggregated SOD1, but that remains to be demonstrated. So basically, that's what we want to do next, and now we are starting the second phase of these two projects. For Peripherin, we will try other antibodies, and we just order them to see what's going on. And the second thing is that we will now use this assay for mutated for samples from a patient with mutation of SOD1, and also later on also look at larger cohorts to see if we can see in the range of the sporadic cases some misfolded SOD using this technology. I thank you for your attention.